Hi, today we're going to talk about tissues. We're going to divide this chapter up into one, epithelial tissues, and next time we will talk about connective tissue. So there are four major families of tissues. Epithelial tissue is what we're going to talk about right now, connective tissue, muscular tissue, and nervous tissue. So different types of tissues will look different histologically. This is epithelial tissue. Let me get a pencil here. You'll notice how the cells are very close to each other. Connective tissue, we'll learn it's kind of like the most variable tissue. Then we've got muscular tissue and nervous tissue. So epithelial tissue in general will cover the body surfaces, outside and also inside the hollow organs. When we talk about the lumen, that is the center, the central part of, like for example, let's say this is an artery. So the center part of the artery is called the lumen. It also lines body cavities, forms glands, and will allow the interaction of internal and external environment. Connective tissue is the most variable. So it can vary from cartilage, which is semi-soft, to bone, which is very hard, to adipose tissue, and also blood. So it can be very, very variable. So connective tissue is considered the glue that connects everything together. Its function is to protect the body. It also supports body and other organs. It helps bind everything together. It can store energy like an adipose tissue, aids in our immunity, and it's very, very variable, like we just described. Muscle tissue we'll talk about in the future. The cells are specialized for contraction. So through these contractions, they need energy and create a force. They assist in moving body and cells. They generate heat. There's three types of muscle cells. There's skeletal muscle cells, smooth cells, and cardiac cells. The nervous tissue is very, very variable. They're very specialized for the transmission of signals. They help us detect changes not only on the inside but the outside of the body, and they generally generate chemical and electrical signals. And we'll talk about this down the road. So we talked about cells in the last chapter. Now let's talk about the contact point between the cells, and generally it's between the plasma membrane tissues. There's 5 types that we're going to talk about. The first kind is called tight junctions. This is a tight seal between these plasma membranes. For example, in epithelial cells that line areas that you want to, for example, hold fluid in, which will include the stomach, the intestines, so it's preventing fluid in these cases to escape. So those are called tight junctions. Adherence junctions has a similar function, but there's a layer of dense proteins that are connected to this. And this we see in the inside of plasma membranes that are attached to both membrane proteins and microfilaments of the cytoskeletons. So these proteins are called cadherins. Desmosomes and hemidesmosomes, these are cell junctions that connect cells to a basement membrane, and also they prevent cells from separating under extreme tension. Like for example, the cardiac muscles. You wouldn't want the cardiac muscles to separate with the contraction of our heart and also some of our skin cells. Hemidesmosomes or half-desmosomes and often are connecting cells to a basement membrane. Gap junctions are these little fluid-filled tunnels where we want to have the passage of either electrical signals or chemical signals. We see this in the nervous system, the heart, and the gastrointestinal tract. So here's some examples of these cell junctions, the tight junctions where the plasma membranes are tightly connected. Adherence junctions are also where we do not want the pulling apart of these cells. So there's a plaque of these proteins called coherins that tightly bond the plasma membranes of these cells. Desmosomes prevent tearing away of cells with activity. Hemidesmosomes, we'll talk about these in a little bit, that are half of these, and they're connecting the cells to a basement membrane. And then gap junctions are these little fluid-filled spaces between the plasma membranes in areas where we have chemical and electrical impulses, for example, in the heart, the cardiovascular system, the nervous system, and also in areas of the gastrointestinal system. So let's talk about epithelial tissue. So when we think of epithelial, it's the layer of cells that forms barriers. There's two types. There's the membranous epithelial tissue, and this is going to cover and lining epithelium. It forms our skin, the covering around our skin, our hollow organs. It's the inner lining of our blood vessels, our ducts, and also our body cavities. We see this in the respiratory, digestive, urinary, and reproductive tracts. Another type of epithelial tissue is glandular. So we'll talk about glandular tissues towards the end of this PowerPoint. And this is areas that secrete hormones like our thyroid, adrenals, sweat glands are all part of the glandular tissue. So when we're looking at the difference between epithelial and connective tissue, you'll notice a big difference. So on this side here, this is epithelial tissue, and this is connective tissue. So as you notice in epithelial tissue, do you notice how the cells are packed tightly to each other? So there's a purpose of this. It's very protective when these cells are packed really close to each other. Connective tissue, you could see the cells are scattered around in what we call a matrix. So this matrix, or extracellular matrix outside the cells, is variable. So for example, in blood, it's going to be fluidy. In bone, it's going to be very, very hard. There's also scattering of these fibers. So again, each of the components within the connective tissue will be characteristic with what kind of tissue we're talking about. So let's talk about the characteristics associated with epithelial cells. So the first one is called cellularity. What this means is that the cells are bound together by a cell junction. Okay. Also polarity. So what does polarity mean? Is that there always is a top surface, okay, and there's also a bottom surface. So this top surface is called the apical surface. Here is an epithelial cell. And the bottom is called the basal cell surface, okay? So there's always a top and a bottom. Attachment means that the basement surface is attached to the basal surface, and they're also attached to each other, okay? Remember, epithelial cells are really close to each other. An important characteristic about epithelial cells are they're avascular. What does that mean? There's no blood vessels there. So let's think of the epithelium of the outside skin. For example, if you have a paper cut, right? It should not bleed, because if it's only in the epithelium, it will not bleed. But does it hurt? Yes, a paper cut hurts a lot. So there are nervous tissue there, but it's avascular. Regeneration means that the apical surface is constantly being replaced through cell division coming from the basal surface. some of the functions that we need to know is epithelium is physical protection. It's protecting and separating the external environment from the internal environment, often from abrasion, dehydration, destruction by chemical and biological agents. Epithelial also controls permeability. So through the protection, it's controlling what is coming through. The level of permeability will be controlled by the type of epithelium that exists. As we talked about, epithelium has lots of nerves, or highly innervated. So it's very important for detecting changes between and controlling like what changes are occurring, separating the internal and external environment through the nervous system. Epithelial tissue also produces secretions through glands. So often these secretions are through physical protection, and also, for example, in blood can act as chemical messengers. So let's look at a model of the typical epithelial cell. So we talked about how this is an epithelial cell. It has an apical surface, which is the top, and this would be the lumen. Okay, so let's say this is, like a gland. This would be the lumen. This is the basement membrane, which is made up of a basal lamina and a reticular lamina. The lateral surfaces are connected. Okay, and this is on the bottom is considered the basal surface. So we talked about how epithelial tissue is highly innervated. You can see the nerves here, but it's avascular. Okay, so how do these cells get their nutrition? They have to survive and get nutrition through epithelial tissue is always connected to connective tissue, which is very vascular. You can see the blood vessel here. So the nutrition from the blood vessel via diffusion is going to enter through the basal surface to give nutrition to these epithelial cells. So we need to learn to classify these epithelium. We need to look at a histological slide and hopefully know three things. First of all, what it's called, generality, where you think it's located, and number three, what type of function that type of epithelium will have. So when we're naming this, we base it on two things. One is lamination. Lamination is how many layers. The second one is going to be cell shape. So when we talk about lamination, or how many layers, we divide it into simple, which includes only one cell, versus stratified. Stratified will be two or more cells. So that's simple, right? Simple will be one cell. Stratified is two or more cells. We'll call it stratified. The second one is versus shapes. So squamous means it's flat, little flat cell. Cuboidal will look like a cuboid or a square, okay? Another way you'll see this described is that it's tall as it is wide. Columnar, or a brick, okay, which kind of will be also described is taller than it is wide. The third one is transitional. What does transitional mean? Transitional means that it will look one way, squamous, and this is mainly referring to the urinary bladder, okay? Urinary bladder, where when the bladder is full, the outer layer will look squamous or flat, but when the bladder is empty, that outer layer will look like a cuboid. And we'll look at these in more detail. So other ways to understand these are the arrangement of cells into layers will also reflect their location and function. So simple epithelium will include one cell. Stratified epithelium will be 2 or more. Then there's another term we have to learn is pseudostratified. So what does pseudo mean? Generally, like a pseudo friend is like a fake, right? So it's looks like it's more than 2 cells, but it's only 1. And the best way is to show you images of this. So we're talking about naming the simple epithelium. You'll see it called as simple squamous. So one little flat cell, simple cuboidal, a series of cuboid cells, simple columnar. Okay, so those are column cells. So we also have to differentiate between ciliated and non-ciliated. So ciliated will be for example, a columnar cell with little like hair-like structures. Okay, so they're cilia, versus non-ciliated will often have what we call microvilli. Okay, so when it's a non-ciliated, often it has these little finger-like projections, and the purpose of this is to increase the surface area. We see these a lot in our GI tract where we want to absorb nutrients. The ciliated, these cilia act like little hair-like structures. We see these often in the respiratory tract or the uterine tubes, and the purpose of these are to collect and trap microbes and dust before it enters the lungs. In the fallopian tubes of the women, these little cilia help at the oocyte or the egg to flow through the fallopian tubes as it reaches the uterus. Of course, if it's fertilized, it will be implanted in the uterus for growth. Pseudostratified columnar is one of these that looks like it's stratified, but it's only 1 cell. So what that's going to look like is all the cells are going to reach the basement membrane, okay, but not all the cells are going to reach the apical membrane. So I'll show you an image of what that looks like. So it looks like it's more than 2 cells, primarily because the nuclei are kind of all over the place. But it is only 1 layer, so we call it pseudostratified. So let's look at these in more detail. So again, we separate these in lamination, which is arrangement of layers. So simple is 1, stratified is 2 or more, right? And then pseudostratified, remember I just talked about how all these cells are connected to the basement membrane, but not all of them are on the apical layer. So it's pseudostratified, meaning it's fake. It looks like it's stratified, but it's not. And the way you're going to tell this histologically is because you're going to see the nuclei all over the place. The other one is the cell shape. Remember, squamous means flat. So this would be simple squamous. Cuboidal looks like a cube. Columnar looks like a column. So we'll learn that whether it's simple or stratified, there's certain functions. A simple one, stratified, you can see it's more in 2 cells. We see these in areas that need more protection, and it's protecting against abrasions, correct? Drying out, infection. So it's a very protective layer because there's lots of cells here, okay? So for example, stratified squamous, okay? We'll talk about this in more detail. Stratified is more than 2 cells. We call it squamous because this top cell here is flat. Do you see that? Even though these look cuboidal, we name these stratified cells based on this apical layer, the top layer. So stratified squamous, we see this in areas, for example, the inside of the mouth, the esophagus, the vagina. So these are areas that need a lot of cells for protection. Stratified cuboidal, so more than 2 cells. We'll see these in the ducts of sweat glands. A function will be secretion. Stratified columnar, more than 2 columns. We see this in the epididymis, in the male, the mammary glands, the larynx, they secrete mucus. The simple squamous are these flat cells. So because it's a very, very thin layer, where do we see this? Is where we have diffusion of molecules. So where are we going to see that? The blood vessels, correct? Where transporting oxygen, also we're taking away CO2, also around the alveoli of the lungs. So this permits the exchange of nutrients, wastes, and gases. Simple cuboidal is a little more protective. So we see this in areas, for example, around our glands and our kidney tubules, where we have secretion and reabsorption of small molecules. Columnar, they're a little thicker. So this is going to be in areas, for example, around our digestive organs. Often you'll see these goblet cells, kind of like a wine goblet, and it's filled with mucus. So let's look at this in greater detail. So the first one was the simple squamous epithelium. Remember, we talked about how it's very, very just a thin tile, right? So that means it's a single layer of flat cells, kind of a tile, like a tile on a floor, right? So mainly we see this in areas of the cardiovascular system, the lymphatic system. This is where the main function is, where we can, through diffusion, cells will pass along very easily. For example, in the blood vessels, the alveoli of the lungs, and this is where we have the function of filtration, diffusion, and secretion. Some terms we have to review are secretion. So what does secretion mean? Is the release of substances like mucus, sweat, or enzymes. Absorption means the intake or reabsorption of nutrients. We talked a little bit about the digestive system. We've discussed these terms in the past. Osmosis is where water molecules are moving from an area of higher concentration to an area of lower concentration without the use of energy. Where diffusion is the area is where molecules are moving from an area of high concentration to an area of low concentration. Filtration is the passage of a filter that prevents the passage of certain other molecules. Like we see a lot of this in our nephrons in our kidneys. So let's see some examples of simple squamous epithelium. It looks like a tile. It has a basement membrane and the apical surface. And we see in areas where we want the passageway of through diffusion in our cardiovascular system, lymphatic system, alveoli. So let's look at the next one. Simple. What does simple mean? Is one. Cuboidal means it looks like a cuboid. So this is a single layer of cuboid-shaped cells. We see these in areas not that typical, lines the ovaries, the lenses of the eye, also kidney tubules, and glands. So this main function is secretion and absorption. So often you could see it in this way. This is of the kidney. Also we see it similar in glands. So the inside here is the lumen. So this is the apical surface of the cell, and this is the basement. And you could tell it's simple cuboidal because you could see all the nuclei that line up around each other. And this is specifically for secretion and absorption. The next one we're going to look at is non-ciliated simple columnar epithelium. So that looks like a column, correct? So non-ciliated means it does not have cilia, but often will have microvilli. Okay, so remember microvilli are those little finger-like cells at the top. So this is the apical surface, this is the basement surface. So we see these in our GI tract or around many ducts. So the function of this one is secretion and absorption. And in the GI tract, we'll also have goblet cells in between here. And remember, we talked about the goblet cells look like wine glasses, and they're secreting mucus for protection and also lubrication in those areas. So non-ciliated simple columnar, you could see the microvilli, okay, which are those little finger-like projections, which increase the surface area of the cell, which is very important in our small intestine of our gastrointestinal system, because that's where most absorption of nutrients occurs. Ciliated simple columnar epithelium. So ciliated, different from the microvilli, is that they're going to look like little hair-likes. So we see these in the bronchioles of our respiratory tract. The cilia traps microbes and unwanted material from going into the lungs. We also see these ciliated columnar epithelium in the fallopian tubes, the uterus, and the paranasal sinuses. The purpose of the cilia is that it's going to trap mucus, and also there's mucus secretions. It traps foreign particles, takes whatever we don't want, and through coughing or spitting it out, eliminate anything from going into the lungs. Also, the cilia helps the oocyte travel through the fallopian tube, and if it's fertilized, implants in the uterus, and if not, will be expelled in the menses. So we could see an image here of the ciliated columnar epithelium. This is in the fallopian tubes. And differentiate that the cilia are going to look like little hair-like structures versus the microvilli are going to look like little finger-like structures. So this is what pseudostratified columnar epithelial looks like. So the pseudostratified, you could see in this image, this is the pseudostratified ciliated columnar epithelium. It has the little cilia, and we call it pseudostratified because it looks like more than two cells, but it's only one cell. And all of the cells are connected to the basement membrane, but not all of them reach the apical layer. And you see the ciliated stratified epithelium primarily in the respiratory tract and the glands. So you can see this one that's located in the glands, but often we will also see it in the respiratory tract. This is what it looks like. Differentiate the cilia and the trachea versus the microvilli in the gastrointestinal system. There is a non-ciliated pseudostratified columnar epithelium. It's not very common. This one you can see in the parotid ducts. So let's review our epithelium that we just talked about. Simple squamous epithelium, one cell, looks flat. We could see this one in areas where we need the transport through diffusion, filtration, and secretion through a very short space. So we see this in the lungs, the heart, the blood vessels, and lymphatic vessels. Simple cuboidal, one cell. We could see this is particularly secretes and absorbs. It's still thin, but a little protective because it's more of a cuboid. And we see these in the ducts of small glands and kidney tubules. Simple columnar, again, one cell. This is the ciliated version, and this is for absorption and secretion. We could see in the bronchi, and also in the uterine tubes of the uterus. Pseudostratified only has one cell. All the cells reach the basement membrane. Not all the cells reach the apical layer. So the cilia in this one, primarily, we can see it in the respiratory tract. And the cilia prevents any unwanted material getting into the lung and also secretes mucus, which also traps bacteria. Now we'll talk about the stratified. So remember, stratified is 2 or more cells. So anytime you see stratified, one of the main functions is always going to be protection, correct? So stratified squamous is very protective from abrasions. We could see this inside the mouth, the esophagus, and the vagina. Stratified cuboidal, not very common. We see a lot of these in the mammary glands. Stratified columnar, again, not very typical, but we see this, for example, in the male urethra, which secretes and protects. Transitional epithelium, we'll look at this in more detail. This lines the bladder, the urethra, and the ureters of the urinary system. And again, we talked about how the top layer will look squamous or flat in a full bladder, and it'll look cuboidal in an empty bladder. So let's look at stratified epithelial tissue. So stratified squamous, we always refer to the shape of the cell based on that apical layer, okay? Because remember, there's more than one cell, so we always name it based on that top layer or the apical layer. So stratified squamous is the flat cell. Stratified cuboidal looks like a cube. Stratified columnar looks like a column. And transitional, depending on if the bladder is full or empty, it could either look squamous or cuboidal, okay? So let's talk about stratified squamous epithelium. So it has 2 or more layers, and we're always naming it based on that apical layer. So stratified squamous epithelium, the non-keratinized form forms the inside of our mouth, our esophagus, and the vaginal and anal areas. And this is mainly for protection from any kind of abrasion, water loss, UV radiation, foreign invaders. The keratin stratified squamous epithelium, keratin is a tough fibrous protein, and we only see this in our skin. And we're going to talk about this to a greater extent in the next few chapters. So the non-keratinized stratified squamous epithelium will look like this histologically. Many cells, that top layer looks flat. The bottom layer is in a continuous state of reproduction, and as it reaches the top, they can slough off, especially in the skin. The non-keratinized form is going to be for protection primarily against abrasion, water loss, UV radiation. Again, we see this on the inside of the mouth, the esophagus, the epiglottis, vagina, all of this region where there's a lot of abrasion. The keratinized stratified epithelium, this is the skin. The keratin is a tough fibrous protein that, in addition, will protect the skin from heat, microbes, any kind of chemical irritants, and we will study this in great detail when we study the skin. Stratified cuboidal, we don't see this that often. It looks like, it's hard to tell here, but there's 2 layers of cuboidal cells, so it's 2 or more cells. We call it stratified. Often we see these in the sweat glands and the esophageal glands and the male urethra. Functions, it's stratified, so 2 more cells, protection, and there's some secretion and absorption. Stratified columnar, again, it looks like a column, not very common. Parts of the urethra and some glands, areas of the anal mucosa membrane also, and the conjunctiva of the eye. And again, this is for protection and secretion. So transitional epithelium is in the urinary tract, and we can generally look at it where when the urinary bladder is full, that top layer will look squamous, and when the urinary bladder is empty, it will look more cuboidal. So that's why we call it transitional epithelium. All right, so make sure you look at all these. I have sent you a handout, and this will help you put it in order, understanding the different types of epithelial cells. So make sure you know 3 things. You could identify the type of cell. Number 2, you could identify a general location. And number 3, a function. So let's talk about glandular epithelium now, which is the second type of epithelium. So this is a specialized epithelial cells to form glands, and they secrete substances either into ducts, the outer surface, or into the blood. And we divide this up into 2. One is the endocrine glands, and the other one is exocrine glands. So endocrine glands are ductless, so they don't have ducts, and they produce hormones. They maintain our homeostasis, and they're putting their secretions into the interstitial fluid or directly into the bloodstream. And this includes our thyroid glands, our adrenal glands, versus exocrine glands. They secrete their product onto body surfaces, and they're the more common ones. Some of the secretions are mucus, sweat, salivary glands, and we'll look at these in more details. So let's look at endocrine glands. So endocrine glands, they secrete hormones, and they're ductless, remember that. And they're secreting their secretions or hormones into either the interstitial fluid or directly into the bloodstream. Some examples will be like pituitary gland, thyroid glands, parathyroid glands, kidney, the adrenal glands that are superior to the kidneys, the pancreas. So these hormones regulate metabolic and physiological activities to maintain our homeostasis. The exocrine glands are the more common ones. They're dumping, they do have gland, they secrete their product into ducts. So they do have ducts, and they're emptying their product to the surface coverings and linings of the epithelium. They can include sweat, oil, earwax, digestive glands, salivary glands. They produce the substances like oil, earwax, saliva, and digestive enzymes. So let's talk about the exocrine glands. So we divide these up either structurally or the classification is structure or function. There's a variety of different structures that we can see in our glands, or the functional classification, which will divide these up into 3 types. One is called the merocrine glands. This is the functional classification of our exocrine glands. So merocrine glands, an example of this is salivary glands. So the way the salivary glands work is that through the whole process of the rough endoplasmic reticulum takes the protein to the Golgi complex, which will produce a vesicle through exocytosis. So it secretes its saliva via exocytosis into the mouth. And that's a merocrine gland. An apocrine gland, an example is the mammary glands, is where part of the cell actually pinches off with the cytoplasm and the cells, and this is what becomes the secretion. So that is mammary glands. And the third one is holocrine glands. So holocrine glands is where the entire cell dies off and becomes the secretory product. And we see this, what's called the sebaceous oil glands. For example, the glands at the base of the hair follicles, which secrete like the oily substance that covers our hair. So again, the merocrine glands is produced, the secretory product is through exocytosis, and one example would be the salivary glands or mucus cells that are secreting their product into the lumen through exocytosis. The apocrine cells is where part of the cell pinches off and becomes the secretory product. We see this in the mammary glands, and we'll see these in apocrine glands in the surface of our skin, for example, what produces body odor. So it's areas, for example, under our armpits, in the genitalia, that secrete like a thick mucousy type of secretion. And it itself doesn't give you body odor, but if left without bathing, after a day or two, it certainly will smell because the bacteria will start to grow on it. The last one is the holocrine glands. This is where the entire cell breaks off and becomes the cell itself. And an example which we'll talk about is sebaceous oil glands at the base of the hair follicles. And that's what gives us the oily secretions at the bottom of our cells. So that is the end today. We are done.